Welcome to the debate. Today we are examining a really profound shift in modern medicine and human biology. We are talking about the massive explosion of AI-driven gut microbiome analysis technology. And to really understand the stakes here, we first have to wrap our heads around the sheer scale of the environment we are talking about. The human gut is this bustling 3-pound ecosystem, home to over 1,000 distinct species of bacteria, viruses, and fungi.
Yeah, and to put that in perspective for folks who might, just view these as passive little passengers, these microscopic entities actually outnumber our own human cells by a factor of 10 to 1. So they are deeply embedded in our physiology.
Precisely. I mean, they influence virtually every system in our bodies. We are talking about foundational functions like basic digestion and immune system training, all the way up to really complex mental health pathways and the development of chronic diseases.
Right.
So our central question today, emerging directly from our source material regarding the science and applications of gut bacteria analysis, is this. Is gut microbiome analysis technology currently a mature, revolutionary tool for global personalized medicine, or do profound limitations in data representation, diagnostic accuracy, and standardization critically undermine its real-world utility?
A big question.
It is, and I will be arguing that plummeted sequencing costs and machine learning integration have already produced actionable, revolutionary healthcare applications ranging from predicting blood sugar responses to actively enhancing cancer immunotherapy.
And I will be taking the position that this field is fundamentally compromised. It has a severe data skew favoring Western populations, major technological limitations in mass market sequencing, and a lack of standardization that just risks generating overblown, non-diagnostic claims.
Let's start by looking at the staggering technological leap we have witnessed here. If you go back to, say, 2003, sequencing a single sample cost millions of dollars. Millions. Today, next-generation sequencing machines can process billions of DNA fragments in parallel for under $100.
The cost drop is impressive, sure.
It changes everything. We are using chemical kits to lyse bacterial cells, extract their genetic material, and then use polymerase chain reaction to amplify specific DNA regions millions of times so we can actually read them. When you target the 16S rRNA gene, which acts like a conserved bacterial barcode, or use shotgun metagenomics, algorithms can assemble a highly detailed microbial map. And when you apply machine learning to those massive datasets, the results are actively solving health crises.
Okay, but look, the drop in sequencing cost is a massive technical achievement. I'll give you that. But we have to look closely at the structural blind spots built into this current ecosystem. The foundational databases used to make these grand AI predictions are severely skewed.
How so?
Right now, only 1% of the samples in these massive reference databases come from Africa or South Asia. 1%. That means the machine learning algorithms you are pointing to are trained overwhelmingly on the microbiomes of Western urbanites.
Well, sure, the databases are growing, but that doesn't negate the immediate clinical utility we're already seeing. Take Day 2's AI, for example. It can predict blood sugar responses to specific foods with 90% accuracy based on gut profiles. 90%. That is a vital functioning tool against global obesity and insulin resistance. Or look at fecal microbiota transplants, FMTs.
Well, FMTs are a very specific.
They are curing severe C. difficile infections with a 90% success rate. We have moved from theory to actionable, life-saving clinical tools.
I won't dispute the targeted success of FMTs in highly controlled clinical settings, but you are describing a mass market revolution built on consumer tests that predominantly rely on 16S rRNA sequencing, and that specific method is fundamentally limited because it entirely misses crucial species-level detail.
It provides a baseline.
You get a broad categorization, not a precise diagnostic picture. Yet these tests are marketed with sweeping health promises. The FDA has already had to crack down on these overblown claims. We saw that with the shutdown of uBiome. We literally have an entire industry sitting on top of low-resolution data sets and, quite frankly, mathematically unsound global extrapolations.
Let's drill down into that resolution argument because it feels a bit outdated. It is true that the earliest iterations of these consumer kits relied on narrower 16S sequencing, but the field is rapidly advancing beyond that baseline. We are now heavily utilizing shotgun metagenomics.
Which is a completely different tier of analysis.
Exactly my point. Unlike the 16S barcode method, which is essentially just scanning the genre of the book, shotgun metagenomics indiscriminately sequences all the DNA in a sample. It reads every single page. That gives us strain-level detail. It identifies the functional genes present, for instance, the precise genes responsible for producing anti-inflammatory compounds in the gut. We aren't just taking a roll call of which bacteria are in the room anymore. We are looking at their actual functional capacity.
And clinically, that is exactly what you need. But let's look at the economic realities laid out in the material. You are pointing to shotgun metagenomics as the gold standard. Scientifically, it is, but a full, accurate metagenomic test currently costs between $200 and $500.
Which is a fraction of what it cost a decade ago.
True, but it still functionally excludes massive portions of the global population from the high-resolution benefits of this technology. The affordable option, the $20 basic kit, is precisely the 16S sequencing that misses the species-level detail entirely. So we have a tiered system. The masses bet low-resolution, non-diagnostic data that paints a blurry picture, while only those who can afford your premium tests get the actual, actionable insights.
I wouldn't call a $20 kit useless just because it isn't shotgun metagenomics. It still provides a vital baseline that literally didn't exist for consumers 5 years ago. Furthermore, companies are moving beyond just DNA altogether. Viome, for example, is using an RNA-focused approach.
RNA analysis is notoriously tricky.
But they are doing it. They are measuring active gene expression. So instead of just seeing what bacteria are present or what genes they happen to possess, RNA analysis shows us exactly what those microbes are doing in real time. We capture the dynamic activity of the gut.
Measuring RNA expression is incredibly sophisticated, yes, but it introduces another major vulnerability: contamination and degradation. Next-generation sequencing is highly sensitive. We are talking about machines using fluorescent markers or subtle electrical signals to identify individual nucleotide bases. Even the absolute slightest contamination can completely ruin a dataset.
And labs have rigorous protocols to handle that sensitivity.
Controlled clinical labs do, yes, but the mass-market ecosystem relies on mail-in kits. We are asking individuals to collect stool samples at home, package them, and mail them across the country or internationally, guaranteeing the highly sterile protocols and temperature controls required for truly accurate sequencing is nearly impossible in that logistics chain. The resolution you are championing with RNA and metagenomics is theoretically possible in a controlled environment, but it degrades significantly in the reality of the mass-market mail-in model.
The logistics of sample stabilization have improved dramatically, using proprietary buffers that lock the nucleic acids in place during transport. But let's look at the broader application of this data, because you mentioned earlier that the databases are overwhelmingly Western, implying this technology doesn't serve the global majority.
It doesn't, not accurately.
I think the successful global adaptation we are seeing directly challenges that notion.
How so? If the underlying catalog is skewed, the outputs must be skewed.
Because the technology is actively being deployed to address localized non-Western biological profiles right now. Let's look at Japan. Firms like Microba are partnering with researchers to link gut profiles to the famous Okinawan Blue Zone diets. They are analyzing how local fermented foods, like miso, specifically boost beneficial Akkermansia bacteria. That correlates directly with their high longevity rates. It is a highly localized application of the technology.
Japan is still a high-income nation with advanced clinical infrastructure. What about the regions representing the massive data gaps, the ones you brushed past earlier?
We see it there too. In Brazil and India, local startups are producing affordable kits specifically to target the rising diabetes epidemics in those specific regions. They are utilizing the technology to address the fact that gut dysbiosis correlates heavily with insulin resistance, a condition affecting 500 million people worldwide. And perhaps most powerfully, look at the nascent scene in Africa. South Africa's Symbiotic Health is using this technology to tackle malnutrition.
But this is exactly where the statistical flaw becomes critical.
Let me finish the example. Their tests have revealed how diverse microbiomes in Ethiopian children actually protect against stunting. This data isn't sitting in a vacuum. It is directly informing UNICEF nutritional programs right now. The technology is already being adapted to map the environmental realities across the globe.
I understand the intention behind those regional initiatives. I really do. But from a purely mathematical and diagnostic standpoint, they highlight the exact problem I'm talking about. When a startup in Brazil or a researcher in Ethiopia sequences a sample, they don't just look at the raw DNA reads. They have to assemble those reads into a microbial map and compare it against reference databases.
Right. Like Green Genes or the Unified Human Gut Genome Catalog.
Exactly. And the material explicitly notes that the Unified Human Gut Genome Catalog, while compiled from over 10,000 global samples, carries a glaring caveat. Only 1% comes from Africa or South Asia. If you take the microbiome of a rural African villager who has a heavily fiber-rich diet and run it through a machine learning model trained on that catalog, the algorithm is fundamentally misaligned.
The algorithm identifies the composition regardless of where the person lives. The DNA is the DNA.
It identifies the composition, sure, but the machine learning model interprets the health risk based on its training data. If it is trained primarily on Western profiles, people eating highly processed foods in sanitized urban environments, it will inherently skew the predictions. It might flag a perfectly healthy, highly diverse microbiome as anomalous or dysbiotic, simply because it mathematically diverges from the gut of someone living in New York or London. Until those foundational reference databases reflect the actual genetic and microbial diversity of the global population, the models will continue to mathematically project a Western biological baseline onto the rest of the world.
That assumes the algorithms are static and totally incapable of learning. The reality is that every time a startup sequences a Brazilian patient or Symbiotic Health sequences an Ethiopian child's microbiome, that new data can be fed back into the system to refine and expand the reference catalogs. The machine learning models are continuously iterating.
They iterate, yes, but the volume of Western consumer data flowing in daily far outpaces the trickle of data from targeted studies in developing regions. The skew is essentially self-perpetuating in the commercial space.
Even acknowledging that the database diversity needs to catch up, we simply cannot ignore the immediate clinical efficacy we are seeing right now. The source material outlines clinical applications that go far beyond consumer diet apps and touch on critical life-saving therapies.
I am certainly open to examining the highly controlled clinical successes, provided we separate them from the wild west of the consumer market.
Fair enough. Let's look at Memorial Sloan Kettering. Researchers there, and in replicated trials in Shanghai, have proven the specific gut bacteria actively enhance the efficacy of cancer immunotherapy. By modulating the gut microbiome, we are literally making advanced cancer treatments more effective. That is a staggering application of this technology.
It is fascinating, truly, and it underscores the absolute necessity of rigorous, controlled science. But how does that translate to the broader commercialization of microbiome science? Because that's what's driving the industry.
It translates because we are decoding the systemic pathways of the body. Take the gut-brain axis. We now know that 95% of the body's serotonin is produced in the gut, mediated by vagus nerve signals. That biological insight has led to targeted therapies, like the Australian startup Seed's DS01 probiotic, which uses global trial data to specifically target anxiety fueled by dysbiosis. And the applications even extend beyond human biology. In Brazil's beef industry, gut sequencing technology is being used to inform animal feed, modulating the microbiome of cattle to reduce methane emissions. That aligns with urgent global climate goals. The efficacy of the sequencing is undeniable across multiple disciplines.
The basic science is undeniable, yes. The trials at Memorial Sloan Kettering show incredible promise.
But when we look at the broader clinical translation across different labs and commercial entities, we run headfirst into a massive lack of standardization.
The industry is relatively young. Standardization takes time.
It does, but right now, that lack of standardization is a critical vulnerability. The source material points out that varying protocols across different companies severely hinder cross-study comparisons. If you take the exact same sample and send it to 2 different labs, you might get a completely different microbial map.
That is primarily an issue of differing proprietary algorithms, not a failure of the sequencing itself.
It is an issue at every step of the process. The extraction chemicals used to lyse the cells vary, the PCR amplification cycles vary, the algorithmic thresholds for identifying a species vary. The International Human Microbiome Standards, the IHMS, is trying to implement universal protocols, but standardization heavily lags behind commercialization. And this lack of standardization creates a massive regulatory and data liability, particularly regarding privacy.
Privacy regulations are evolving alongside the technology. GDPR in Europe is already actively applied to this data.
The regulations exist, but the inherent nature of the data makes the risk profound. We are not just talking about a list of stomach bugs here. This is highly sensitive biometric data. The material clearly states that your gut data can reveal your ethnicity or your predisposition to specific disease risks. Because the data is so revealing, stockpiling it in commercial databases is a massive liability.
Which is exactly why we're seeing regulatory enforcement. It isn't a free-for-all.
Getting hyper-personalized insights. We have consumer apps like Zoe in the UK pairing sequencing with continuous glucose monitors and wearables. Or China's iGut integrating with massive platforms like WeChat for real-time lifestyle advice. That is an enormous amount of highly sensitive biological data flowing through commercial tech platforms that lack universal standardization. Without ironclad, standardized data frameworks, this isn't just a clinical diagnostic tool, it is a sprawling data liability. Exposing an individual's ethnic background or future disease risk through a mail-in stool test is a structural flaw the industry hasn't solved.
The data privacy concerns are real, but they are not unique to microbiome analysis. We face the exact same concerns during the rise of human genomic testing, and we successfully developed frameworks to manage those liabilities. In Europe, GDPR-compliant services are already proving that you can prioritize user privacy while still feeding anonymized, vital data into massive scientific endeavors.
Anonymizing genomic data is notoriously difficult, and the microbiome is just as unique to the individual, if not more so.
But the benefit of aggregating that data securely is immense. This data is contributing to EU-wide studies like the MetaHit project, which has sequenced over 3,000 Europeans to uncover the precise triggers for inflammatory bowel disease. By analyzing this data at scale, we are identifying exactly why IBD is rising in Europe and Japan, and why allergies are spiking in urban China. We are identifying low bifidobacterium levels and testing targeted interventions with prebiotic fibers and probiotics. The actionable insights we gain from optimizing the recovery of athletes using butyrate-producing microbes to managing type 2 diabetes globally far outweigh the regulatory growing pains.
The science of butyrate producers and bifidobacterium is sound, but when dealing with human biology, the application has to be diagnostically robust, not just suggestive. We can identify that an elite marathon runner has a certain gut profile that favors butyrate producers, which aids in recovery. That's great, but that does not mathematically mean that selling a consumer a probiotic pill containing those microbes will yield the same athletic recovery.
It provides a targeted baseline for improvement rather than just guessing.
But correlation is not causation. Aside from highly regulated, specific procedures like FMTs for C. difficile, the vast majority of what is being marketed to consumers operates in a gray area of wellness rather than regulated medicine. The FDA shutting down Ubiome wasn't a minor regulatory hiccup. It was a fundamental reckoning for an industry that routinely promises diagnostic certainty based on exploratory data. Until we standardize the varying protocols so that a test in Mumbai yields the exact same baseline metrics as a test in Silicon Valley, we are building a multi-billion-dollar industry on an unstable foundation.
I disagree that the foundation is unstable.
Even if it doesn't meet the strict criteria of a traditional diagnostic tool in every single consumer application, the utility of predictive modeling is incredible. A system like Day2's AI empowers users to make hyper-personalized dietary choices that actively prevent the onset of severe metabolic conditions. Moving away from generic, one-size-fits-all dietary guidelines to insights based on your unique microbiome ecosystem is a true paradigm shift in preventive medicine.
Predictive modeling is only as mathematically sound as the data it relies on. If the models continue to learn from populations with entirely different environmental exposures, diets, and genetic backgrounds, the predictions remain statistically flawed for the global majority. The geographical data gaps have to be closed, and the extraction and sequencing protocols absolutely must be standardized before we declare this a global medical revolution.
As we draw this discussion to a close, I want to summarize my position based on the evidence we have explored. The transition from the early 2000s to today has been a monumental leap, driven by plummeting costs in next-generation sequencing, processing billions of reads for a fraction of the historical cost, and the integration of sophisticated machine learning. Microbiome science has indisputably moved from theoretical research to actionable therapy. From mapping active gene expression with RNA analysis to the 90% success rate in curing C. difficile to actively enhancing cancer immunotherapies, the applications are profound. There are undoubtedly growing pains regarding database representation and data privacy frameworks, but the technology is already providing hyper-personalized insights that tackle global health crises like obesity, diabetes, and malnutrition.
And to summarize my stance, I maintain that while the scientific potential of the microbiome is vast, the current commercial and technological execution is deeply flawed. Until the massive geographical data gaps, specifically the mere 1% representation from Africa and South Asia in foundational catalogs, are closed, the machine learning algorithms will remain statistically skewed. Furthermore, until the industry moves definitively past mass-market 16S sequencing limitations, solves the contamination risks of mail-in logistics, and adheres to stringent, universal standardization through bodies like the IHMS, the field risks selling overblown promises. The liabilities of exposing disease risk and ethnicity further compound these structural vulnerabilities.
Despite our different perspectives on the current maturity and execution of the technology, I believe we both strongly agree on one fundamental point. The gut microbiome is undeniably a crucial, central driver of human health.
Absolutely. There is no question that this complex ecosystem of bacteria, viruses, and fungi plays a starring role in our biology. Pairing machine learning with DNA and RNA sequencing has provided an unprecedented window into human physiology.
Understanding both the staggering potential of this personalized medicine and the serious risks of relying on statistically skewed biological data sets allows us to form a much more sophisticated view of this quiet revolution in the human gut. There is so much more to explore in the source material regarding how these microscopic ecosystems will shape the future of global health.
The science is moving incredibly fast, and the implications for everything from mental health to chronic disease are vast and complex.
Thank you for joining us for this rigorous exchange of ideas. We leave it to you, the listener, to weigh the incredible breakthroughs against the structural challenges and form your own conclusion.